All right, welcome to chapter 13, where we are comparing two means and two proportions. It's two sample problems. And the goal is to compare the responses to two treatments or two populations. So if you remember, we talked about the difference between random assignment to treatments. This is what you guys do in experiments, or you can do random selection from populations. Okay, so you might take the population of males and the population of females. You might take the population of Massachusetts and the population of Connecticut. You might take the population of Concord Carlisle High School and the population of Lexington High School, right? Like, we can compare two different populations where we take a random sample from two different populations and compare them, or we can randomly assign them to two treatments and compare them. Okay, and so this is what most of you guys will be doing for your inference on your projects. So we have a separate sample from each treatment or each population, and the responses of each group are independent of those of the group. So independence, we see there can't be interaction between the two groups. All right, and so we want to have independence. So comparing the two means: simple random sample or simple or random assignment. Okay, we need normality. Both populations are normally distributed. Okay, it's enough, it says here, that they have similar shapes. So like, if you imagine, we're going to end up subtracting the two. So if one's shaped like this and the other one's shaped like this as well, then when I subtract them, the stuff here is going to cancel out with each other, and it's going to lead to, so if I do that minus that, it will lead to something that's normal-ish in the sampling distribution when we combine them. So that's part of the reason why it says if they have similar shapes and there's no strong outliers, then the means and standard deviations. This is for unknown, so we're going to see that it's going to be a t distributions, okay? Because we don't know standard deviations. So independence, samples are independent, and they're independent of each other, and they're independent of that they're not exceeding the population, okay? So this is what we're doing inference on. We have two means, and we're wondering, are these means the same or different? So like, do the two populations or the two treatments have the same mean? And what we compare, this is what we compare, okay? We compare the means of my samples to make an inference about the means of my population, okay? And so we need to look at the difference between these means. So here's an example problem, okay? Does increasing the amount of calcium in our diet reduce blood pressure? So they did this. They looked at the relationship between calcium and intake and blood pressure for black men, okay? So 21 healthy black men, randomly chosen, 10 received a calcium supplement and the other received a placebo. Okay, the experiment was double blind, so they didn't know whether they were receiving it, and also the people who were interviewing them and asking about their data. Okay, so they measured the systolic blood pressure after 12 weeks. Okay, and this is what we saw. Say group 1, here's the change that they experienced. Group 2, here's the change that they experienced, and we're hoping that it reduced the blood pressure. So we're looking for negative numbers, okay? So, or this is the reduction in blood pressure. So here, this says they went down by 5 on average, and there's the standard deviation. In this group, went actually up, but minimally when they were receiving a placebo, and there's the standard deviation, okay? So what we need is randomness, okay? Did they randomly assign these people? Yes, this one's random assignment to treatments. Are they independent of each other? Yeah, it's not like these people were necessarily like interacting with each other. I'm sure there's more than 210 healthy black men. And is it going to be relatively normal? So for this, I don't know if these are relatively normal. And so what we'd want to do is look at some sort of plot or picture to talk about normality, okay? But this is the stuff we would need to run our inference. So if you look at these plots, okay, this one is the placebo group, it looks relatively linear, okay? Probably no outliers, but this is relatively normal. This one actually has quite a bit of curve, but it's not strong skewness or outliers. We're fine, okay? And so you look at these two and you just say, no strong skewness or outliers, okay? And then you proceed with caution, okay? So those are the conditions we need for inference, and we'll talk more in the next video about the actual test statistic and performing the inference. So we'll see you in the next one.